Guys. Just need a brief sip of water there. So let's continue on. So this is the difference between the homologous pairs, which again, we're going to get one from your maternal lineage and one from your paternal lineage. So they're going to have the same genes, but potentially different alleles, whereas sister chromatids are going to be exact copies of one another. So they're going to have the exact same alleles the entire way down. And when the maternal and the paternal homologs cross over, we're going to have exchange of genetic information. And this happens about 3 times per chromosome. So really what happens is your mom and your dad shuffle information before they even create you. And that happened with your grandparents and with those grandparents and with those grandparents, meaning that the DNA in your body is actually a genetic combination of all of your ancestors. All right, so the orientation of the homologous chromosomes during metaphase 1 is a complete matter of chance. It's like flipping a coin. You could be left-right or you could be right-left. And that's called independent assortment. And if we do this independent assortment event for all 23 chromosomal pairs, that's going to give us an 8 million different options of how we could independently assort each of our chromosomes just from one individual without recombination. Okay, so one of the 2 major things that are cool about meiosis, that are unique and not found in mitosis, is the fact that we have something called synapsis. And during this time frame, homologous chromosomes are cinched together down their entire length, so crossing over can occur at multiple locations. And something called reduction division, right? So we have 1 replication event and 2 division events. So 2 nuclear divisions and only 1 replication. So that means that the amount of genetic material is actually halved. That means that we're going to end up with haploid cells. And this is cool. So it leads to what's called, again, sexual reproduction. It's a really neat way that we can end up with genetic variation because we end up with constantly new genetic combinations by these recombination events. And the 3 major mechanisms that help produce the variety that we see is that independent assortment, again, whether our coin flip is going to be left-right or right-left with the chromosome 1, same thing with chromosome 2, same thing with chromosome 3. The crossing over event, and then random fertilization. If you think just from your parents alone, we have over 8 million different sperm competing to fertilize in the egg. And that's not factoring in the fact that there are over 7 billion people on the planet that could be the combination of sperm and egg. So you really are very, very unique and very lucky to be you. Okay, these last 2 slides here are a review for study purposes, the difference between somatic cells and germ cells, what type of cell division is found where, the purpose, whether they're diploid or haploid, and here's the answers for the review. So take some time to go through all of that, and I appreciate you coming to class, as always. I hope you guys have a very fantastic day, and as always, happy studying. Take care, and bye-bye.